So let's start today's PT 365 discussion on science and tech. It is published via Vision IAS Institute. So let's instantly dive into the topic. First topic is related to genes and the concepts. So genes are the segment of DNA. DNA का एक छोटा पार्ट को जीन बोलते हैं and DNA is an important nucleic acid that is found in human cells. DNA is also present with the RNA. And chromosomes are thread-like structure made up of protein and is a single molecule of DNA. Then chromosomes are the one in which DNAs are stored. Then genome is the entire set of DNA. If we take out the DNA and spread it all across a path, then it's all this set of DNA will be genome. Then a comparison between DNA and RNA is that is DNA is double stranded. It is double stranded, like it will look like this. RNA is single stranded, it means it is like this. Then nitrogenous base that DNA has is ATCG, adenine, thymine, cytosine and guanine. Then RNA has uracil instead of thymine. Instead of thymine here, RNA has A, uracil, cytosine and guanine. फंक्शन क्या होता है इसका डीएनए का? स्टोर्स जेनेटिक इन्फॉर्मेशन फॉर इनहेरिटेंस. आरएनए फंक्शन का आरएन का फंक्शन होता है फॉर द बेसिकली फॉर द प्रोटीन सिंथेसिस. देन व्हाट इज genome editing? It is also known as gene editing. It enables scientists to change organism DNA, leading changes in physical traits, eye color and disease risk. Uses site-directed nuclease (SDNs) to make changes that may either be small deletion or a substitution or addition. इसमें क्या है genome editing में? Basically, we use this SDN formula or a nuclease to change a small part of DNA or to substitute a small part of DNA that is causing any defect in a human being with other DNA that is fit DNA. Either we can substitute it or we can add to a DNA or we can delete some part of DNA using SDNs. Nuclease basically. SDNs refers to the practice of cleavaging. Cleavaging DNA strands means cutting DNA strands. If a DNA strand is like this, then cutting few part of it. Then we have gene editing tool. That is the first and the major gene editing tool is CRISPR-Cas9. Then second we have is zinc finger nuclease. Third we have transcription activators like talins. These activators basically help in activating the genes or any enzymes. Then genome editing is different from genetic engineering. In that sense, the genome editing does not involve introduction of foreign genetic material. See, genome editing में हम foreign genetic material नहीं लाते हैं. And genetic engineering में we do introduce a foreign genetic material. For example, genetic engineering the example of it would be GM mustard, in which we are actually adding some bacterial genome into the crop. That is, adding foreign genetic material in it. Then talking about CRISPR-Cas9, it changes the genetic code or edit DNA at a particular location. किसी एक location पे ये change लाएगा. Then we have how it works. It works as cut and paste mechanism on DNA strand. If it is a DNA strand, यहाँ से अगर हम कट करेंगे तो यहाँ पे इस प्लेस पे हम कोई और स्ट्रैंड को लाके पेस्ट करेंगे. जहाँ पे हमें इसको पेस्ट करना होता है ना, दिस शुड बी आइडेंटिफाइड. तो दिस CRISPR-Cas9 genome editing tool do identify the small part and then it performs whatever duty it has to perform there. Cas9 protein is used as a pair of molecular scissors to cut apart. This Cas9 protein will cut apart. This cutting would be done by Cas9 protein. Then genome sequencing. Genome sequencing means determining the exact order of the base pair of individual strand. Base pair means what we have like A, T, C, G, how these base pair are arranged in a DNA. अगर हम इसको एग्जैक्ट ऑर्डर में लिख लें पूरा तो दैट वुड बी जीनोम सिक्वेंसिंग. एप्लीकेशन ऑफ जीनोम सिक्वेंसिंग में क्या हो सकता है, पेटर्निटी टेस्ट. लाइक अभी जो सबसे ज़्यादा चलता है वो पेटर्निटी टेस्ट होता है कि हाँ इफ़ ही इज़ द फादर ऑफ दिस चाइल्ड और शी इज़ द मदर ऑफ दिस चाइल्ड और नॉट. देन इन एग्रीकल्चरल एरिया इज ऑल्सो वेयर वी कैन आइडेंटिफाई द ट्रेड्स फॉर हायर ईल्ड. जिससे ईल्ड बढ़ जाए, डिजीज रेजिस्टेंस हो जाए, क्लाइमेट रेजिलियंस आ जाए, हम ये चीज़ ऐड कर सकते हैं या फिर फाइंड आउट कर सकते हैं कि विच जीन इज रिस्पॉन्सिबल फॉर दैट. यहाँ पे प्रिडिक्टिव डायग्नोस्टिक भी हो सकता है. इट मीन्स कि हाउ प्रिडिक्टिव डायग्नोसिस मीन्स कि इफ अ चाइल्ड इज इन अ वूम हाउ ही इज गोइंग टू बी लाइक हेल्दी और अनफिट, काइंड ऑफ दीज थिंग्स. देन we have Nobel Prize in Medicine 2024. It was also related to gene regulation. Nobel Prize went to Victor Ambros and Gary Ruvkun for discovery of microRNA. You have to remember discovery of microRNA and its role in post-transcriptional gene regulation. I don't have that much of idea on this topic, but yes, what I have to remember or you guys have to remember is who got the prize and for what. It is for microRNA. You have to remember this term, microRNA. Okay, nothing much more in this. What about microRNA? What is it? It is a small non-coding RNA that helps cell regulate gene expression. Controls gene expression by binding with mRNA. mRNA is different from microRNA and preventing them from being translated into proteins or by degrading or destroying mRNA altogether. See, इसमें क्या होता है ना कि this microRNA will bind with mRNA. तो ये mRNA के साथ अगर ये बाइंड हो जाता है ना तो इट विल प्रिवेंट mRNA टू ईदर मेक प्रोटीन, जो उसका फंक्शन है हमारे आरएनए का जो फंक्शन होता है मेकिंग प्रोटीन, या तो उसको ये काम करने से रोकेगा या डिग्रेड होने से mRNA को रोक देगा या फिर डिस्ट्रॉइंग mRNA ऑलटुगेदर. ये तीन काम ये प्रिवेंट कर देगा तुम्हारा माइक्रो आरएनए. Then we have gene regulation. It is the process that control the timing, location and amount in which genes are expressed. तो ये चीज़ है जीन रेगुलेशन. Human organs and tissue consist of different cell types. The chromosomes in the cell contains same set of genes with the same set of instructions. However, due to gene regulation, अगर हम gene regulation करते हैं तो these different cells express unique set of proteins enabling them to perform a specialized function. हम थोड़ा सा इसमें अगर कुछ edit कर देते हैं या फिर हम कुछ add on substitute कर देते हैं तो the same function that the cell has to perform would change. Then we have reverse transcriptase. This concept is important. Researchers have found that when infected by virus, bacteria use RNA to bind the reverse transcriptase. Reverse transcriptase means they are RNA dependent DNA polymerase, a group of enzymes that plays a unique role in flow of genetic information. These enzymes enable the reverse transcriptase reaction. The reverse transcription is synthesis of DNA from an RNA template. Transcription means RNA से DNA बनेगा. Reverse transcription means DNA से RNA बनेगा. From RNA DNA बनेगा. And next we have is genetically modified crops. Okay, so Supreme Court has said a split verdict on the validity of union government decision to grant approval for environmental release of GM mustard crop in 2022. GM mustard crop क्या है? DMH इसका scientific name है 11. It has been developed by Center of Genetic Manipulation of Crop Plants, University of Delhi. GM mustard had not been released for commercial cultivation yet. We have to remember this point. It has not been released for commercial cultivation. It is the India's first indigenously developed GM crop. This is also important. First indigenously developed GM crop. DMH11 is a result of crossing. It's a cross between two mustard varieties, that is Varuna, and one East European, that is early Hira variety 2. This cross has done after introducing a Barnase and Bastard gene. This Barnase and Bastard gene from soil bacterium known as Bacillus amyloliquefacinus. So this is the bacteria from which we have derived these two genes, that is Barnase and Bastard gene, and these genes have been introduced in this DMH11 crop that is again produced via crossing between two mustard varieties, that is Varuna. It is the indigenous variety of India, and the European variety, that is early Hira 2. And the bacteria is obviously Bacillus amyloliquefacinus. Then other GM crops in India is BT cotton. BT cotton has been commercially sanctioned. It is non-food and only approved GM crop in 2002 for commercial cultivation. Introduced to protect against widespread bollworm such as pink bollworm disease से pest से बचाने के लिए we have introduced this BT cotton. It is the first non-food and only commercialized GM crop. Then we have BT brinjal also. 2009 में it was cleared by GEAC body for commercial cultivation, but it is put on a moratorium. It means it has put on a held now. बेनिफिट क्या होते हैं? हायर ईल्ड हो जाएगा, पेस्टिसाइड्स का यूज़ कम हो जाएगा, न्यूट्रिशन वैल्यू ऐड कर सकते हैं हम GM क्रॉप्स में. फॉर एग्जांपल देयर इज गोल्डन राइस विच हैज़ बीन गिवन इन PDS सिस्टम सो दैट न्यूट्रिशनल वैल्यू हम PDS में भी बढ़ा सकें. टॉलरेंस टू ड्रॉट. देन रेगुलेटरी फ्रेमवर्क क्या है? फर्स्ट ऑफ ऑल दिस फसाई एक्ट ऑफ 2026 prohibit import, manufacture, use, sale of GM without FSI approval. Then there's a review committee on genetic manipulation under the Department of Biotechnology. Reviews various aspects of R&D projects involving GM organisms. Then we have State Biotechnology Coordination Committee. It also reviews the safety and control measures. Then we have district level committee. We have state level, and then we have district level committee. Then we have this committee, Genetic Engineering Appraisal Committee. It is important. So this is a statutory committee. It means it has been made by a law. It has been constituted under a law which reads as rules for manufacturing, use, import, export, and storage of hazardous microorganism and genetically engineered organism or cell in 1989. These rules were framed under the Environment Protection Act 1986. So the ministry in which GEAC is MOEF and CC. It means Ministry of Environment, Forest and Climate Change. Responsibility is obviously about safety measures and all, preventing large scale hazardous use, proposals relating to approval and all. Then we have heritable human genome editing. South Africa becomes the first country to allow heritable human genome editing. You have to learn this. South Africa. Unlike somatic cell editing, which affects only individuals, somatic cell में अगर हम edit करते हैं तो it will affect the only individual in which the cells have been edited. It will not pass to his progenies. But in heritable human genome editing, as the name suggests, changes in the germline cell. I mean the cells which are responsible for inheritance, वहाँ पे changes हो जाएंगे. That is sperms, egg or embryos, enabling the inheritance of the trait by the offspring. इसका application क्या हो सकता है? Disease prevention obviously, assisted reproductive technology, for example in vitro technology. Concerns क्या होता है? Unforeseen consequences, because this has been in early development force ना, तो हम अभी इसका consequence उतने अच्छे से read नहीं कर पा रहे हैं. So there are some unforeseen consequences. Then there is societal impact, creation of designer babies. Yes, trait like मतलब people can modify the intelligence level or whatever physical trait they want. So these come under designer babies. We have RNA editing, the first successful clinical demonstration of RNA editing in human was conducted. Wave Life Science has successfully performed RNA editing to treat alpha 1 antitrypsin deficiency, an inherited disorder. This alpha 1 antitrypsin deficiency is inherited disorder, and RNA editing for this has been conducted by Wave Life Science. Wave Life Science. In ATD, the proteins in alpha 1 antitrypsin builds up and affect liver and lung. It affects liver and lung. RNA, what is RNA editing? It is a process that modifies genetic information on RNA sequence through insertion, deletion, or substitution. The scientists use technique called adenosine deaminase acting on RNA, that is ADAR, to guide RNA. Then this gRNA are small RNA molecules that direct editing machinery by base pairing with mRNA in specific regions for modifications. This gRNA will help कि कहाँ पे हमें modification करना है in RNA molecules. Then comparison between RNA and DNA editing is form of change. DNA editing make permanent change and RNA editing will make temporary change. RNA editing is safer and flexible while DNA editing is irreversible. Allergic and immune reaction. DNA editing has higher risk of undesirable reaction as compared to RNA editing. DNA editing tool use protein from certain bacteria to perform cutting function, while RNA editing relies on ADAR enzyme already present in the human body. I mean DNA editing होता है तो हम उसमें जो चीज़ प्रोटीन यूज़ करते हैं फॉर कटिंग द DNA सीक्वेंस, जहाँ पे हम दूसरा DNA लाएं या फिर सब्स्टिट्यूट करें या फिर उसको डिलीट करें, वो चीज़ आती है फ्रॉम सर्टेन बैक्टीरिया, एंड obviously introducing certain foreign bacteria in body will have a disastrous effect in body, because body का भी खुद का self defense mechanism है. It will be activated. It will not allow that bacteria to do its job. While RNA जो enzyme यूज़ करता है वो ADIR है जो body में already present होता है. तो body में present है, it means it's not foreign. तो body will allow it to work as a desired, whatever desired function it has to perform. Then we have bridge recombinase mechanism. Scientists have discovered a naturally existing DNA editing tool. Naturally existing. It's not designed, it is naturally existing. Bridge recombinase mechanism which utilizes mobile genetic elements or jumping genes. These genes are like they jump from this DNA to that DNA. ये चीज़ हो जाता है इनका jumping, इसीलिए इनको बोला गया because they shift their places. BRM, the extra DNA at the ends of jumping gene gets joined together and convert that DNA double helical structure into single RNA, single stranded RNA. ये double stranded DNA को single stranded RNA में convert कर देता है. This bridge RNA molecule can bind two DNA segment, donor and target, allowing for flexible DNA modification. Jumping, what is jumping gene? Jumping genes are DNA sequence that move from one location on the genome to other. Genome मैंने बताया था कि अगर आप सारे DNA को ऐसे रख दोगे लाइन में तो वो जो सीक्वेंस बनेगी that is genome. तो वही jumping genes are DNA sequence that move from one location to, ये अगर यहाँ पे एक जीन है तो वो यहाँ से उठ के यहाँ पे आ गया तो it has shifted its location. That is what is known as jumping place. That is why they are called jumping gene. These are known as transposable element because it can transport, it can change its place. Then these small DNA segment contain recombinase enzyme along with extra DNA segment at the ends of gene that bind and manipulate DNA. They can replicate themselves and insert copies at new location. Their movement can cause genetic mutation and contribute to genome evolution. Then we have one day, one genome. What is it? The Department of Biotechnology and BRIC has launched One Day, One Genome initiative on first foundation day of BRIC. What is BRIC? Biotechnology Research and Innovation Council. इसके foundation day पे One Day, One Genome initiative has been launched by Department of Biotechnology along with the BRIC. What is the aim? Aim is to annotate microbial genome, will be publicly released every day to make microbial genomics data. सी जहाँ पे ना माइक्रोबियल जीनोमिक्स का डेटा एवरीडे बेसिस पे अपलोड कर दिया जाएगा सो दैट वी कैन ट्रेन एनी एआई मॉडल और वट एवर मॉडल दैट हैज बेसिकली इट्स अ डेटा बेस. It will highlight the unique bacterial species found in the country. Then, what is microbial genomics? It is a scientific field that studies complete genetic material of microorganism to understand their structure, function, evolution, and interaction with other organisms. Microbes are too small to see with naked eyes, for example, bacteria, algae, and etc. Significance. What is the significance of studying microbe? First of all, human health, understanding relationship between disease causing bacteria and their genetic makeup. Then we have advancing biotech applications, environment sustainability, human health and disease management, and then lastly in agricultural. For example, how nutrient cycling is happening, nitrogen fixation, maintaining soil fertility, and etc. Other developments related to this genome and genes topic is first of all recombinant protein. IISc has developed new process for production of recombinant protein. What are recombinant proteins that are artificially produced by genetic engineering technique. Artificially produced. They are not naturally occurring. Artificially produced by genetic engineering technique. These are modified to manipulate proteins encoded by recombinant DNA. Then rDNA is artificial. See, wherever R is written recombinant, it means artificially. So rDNA is artificially made DNA strand that is formed by combination of two or more DNA molecules. rDNA technology is used to combine or transfer DNA from one species to create genes with new functions. Production. Recombinant protein such as monoclonal antibodies are mass produced by growing modified bacterial and viral or mammalian cell in large bioreactors. Then these MABs like for example monoclonal antibodies are lab made proteins mimic natural antibodies. इसका एप्लीकेशन ऑब्वियसली इन बायोथेरेपेटिक्स होगा, वेक्टर वैक्सीन्स बनाने में होगा, एग्रीकल्चर में होगा, इन्वायरमेंट में होगा. देन वी हैव टेमेस्टेरस oblanceolate, fern with largest genome. Okay, this is the fern with largest genome. इसका scientific name थोड़ा सा अतरंगी है. Mesipteris oblanceolate. Okay, T must be silent here. Largest, you have to remember this is a fern that has the largest genome. Contains 160 billion base pairs. And ये 50 times larger है human genome system से. It is a plant, group of plant that has pre-model means that has evolved before dinosaur came to earth. So then next is DNA repair. Recently scientists have discovered a new target for cancer treatment which is used by cancer cell to regulate DNA repair during cell division. Cancer cells uses enzyme like TDP1 to repair DNA damage caused by chemotherapy like camptothecin, leading to treatment resistance. TDP is an important enzyme in humans that play a crucial role in DNA repair. So basically, who does the DNA repair? It is TDP1. That is what you have to remember. It is in humans by removing damaged DNA bases. It is a mechanism that helps the cell to maintain its integrity. It exists in both prokaryotic and eukaryotic organism. जहाँ DNA repair दोनों में exist करता है, prokaryotic and eukaryotic organisms में. Plants, plant genome editing tool. अभी हमने बात करी थी CRISPR-Cas9, that is for human. अगर plant की बात करें तो it is called as ISD-RA2-TNPB. And it has been developed by ICAR, Indian Council of Agricultural Research. Miniature gene editing tool, that is ISDRA2TNPB. अब लास्ट का जो TNPB है, इट्स अ प्रोटीन considered the evolutionary ancestor of Cas12 nucleus. Now about this tool is, it is derived from bacteria called Deinococcus radiodurans, survive in extreme environment conditions. Do remember the bacteria name, it is Deinococcus radiodurans. And what editing tool has been made? It is ISDRA2TNPB by ICAR. That DNA segment is also belonging to a family of jumping genes. Significance क्या होगा TNPB का, कि टारगेट यूनिक रीजन इन द डिनोम जो Cas9 और Cas12 नहीं कर पाएंगे, वो TNPB कर लेगा प्रोटीन. बाकी not needed. Then what is exosomes? An exosome based genome editing platform, that is safe exocas. ये भी जीनोम एडिटिंग का प्लेटफॉर्म है. हैज़ बीन डेवलप्ड बाय साइंटिस्ट एट कोलंबिया यूनिवर्सिटी. सेफ एक्सोकैस हैज़ बीन डेवलप्ड बाय कोलंबिया यूनिवर्सिटी. What are exosomes? These are naturally occurring vesicles that have potential to be manipulated to become promising drug delivery vehicles. See, ये vesicles होते हैं, naturally occurring होते हैं, पर अगर इनमें हम थोड़ा सा मैनिपुलेशन कर दें तो ये ड्रग डिलीवरी एजेंट्स बन सकते हैं. They can deliver the targeted drug to targeted location effectively. Exosomes are membrane enclosed vesicle. I mean, ये जो अगर कोई वेसिकल है तो इनका ऐसे आगे मेम्ब्रेन होगा, उसके अंदर ये इनक्लोज होंगे. रिलीज इनटू द एक्स्ट्रा सेलुलर स्पेस, जो भी सेल में अगर कहीं स्पेस है तो वहाँ पे ये रिलीज हो जाते हैं वेसिकल्स, एक्सोसोम्स. नेक्स्ट वी हैव जीन थेरेपी. Gene therapy is a technique that uses gene to treat, prevent or cure a disease or medical disorder. In the most gene therapy, a normal gene is inserted into the genome to supplement the abnormal disease causing gene and restore the target cell to normal state. See, यहाँ पे सेल विद नॉन फंक्शनल जीन है. हमने यहाँ पे एडिंग डीएनए, हमने एक डीएनए स्ट्रेन को यहाँ पे ऐड करा. नाउ द सेल इज फंक्शनिंग प्रॉपरली बिकॉज जो डीएनए खराब था सेल में, उसकी वजह से सेल वॉज नॉट परफॉर्मिंग वेल. नाउ वी हैव वाया जीन थेरेपी वी हैव एडेड अ डीएनए कंटेनिंग फंक्शनल वर्जन ऑफ द लॉस्ट जीन. जो भी जीन नहीं है उसका फंक्शनल वर्जन डीएनए हमने डाला इसमें. नाउ द सेल इज परफॉर्मिंग गुड. It uses a product method such as plasmid DNA. It uses plasmid DNA. Types of gene therapy, there is germline gene therapy. In germline cells like eggs and sperm में modification होगा. Then there is somatic cell gene therapy. Somatic means other than germline. वहाँ पे होगा somatic cells में. अगर germline में gene therapy होती है तो that would be inherited by the offspring. एंड अगर सोमैटिक सेल में होता था वो सिर्फ उसी इंडिविजुअल के लिए होगा जिस इंडिविजुअल में ये सारे थेरेपीज हो रहे हैं. हाँ, नाउ जीन थेरेपी कुड बी यूजफुल फॉर सिकल सेल डिजीजेज, हीमोफिलिया ए, ल्यूकेमिया एंड ऑल. देन वी हैव काइमेरिक एंटीजन रिसेप्टर car T cell therapy. India's first homegrown gene therapy, that is next CAR-19 CAR T cell therapy for cancer has been launched by President of India. Okay, next CAR-19 CAR T cell therapy is indigenously made in India, has been launched for cancer treatment by President of India. Developed by IIT Bombay. Then we have about this is modifies immune cells, specifically the T cells, by turning them, they modify this T cells and prevent them. They turn them into cancer fighting cells known as CAR T cells. T cells are special cells. It is a type of white blood cells. Primary function इनका होता है कि killing other cells. So by making them potent so that they kill cancer cells, हम कैंसर को prevent कर सकते हैं कैंसर को. T cells are taken from patient blood and are changed in lab by adding a gene for man-made receptor called CAR. CAR are protein that assist T cells to recognize and attach with specific proteins present in the cancer cells.